Yeah, so I gotta check around that.
All right, we're set. We're set.
That's cool.
We've got.
So there's many people.
Sorry.
So I found out maybe like a few weeks ago, I had like a flat bag.
That's
And
awesome.
it doesn't like a lot, but there was probably like hundreds of thousands.
Yeah, I did that over in what was it?
We like
Or Red Top Mountain. So I kind of were in the room with guys and we just like kind of across the shoulder they went shop. It's fun. Yeah, but that's good to know. You're like I'm like what, like 5-6 minutes away from the town lake. So that's good to know. Yeah. So have you what was it like in cultivation? Have you messed with I might get the name up, but I think it's the plasticating mushrooms, like Pestalogeosis microspora and stuff. Okay. I tried messing with that one. I could not get it.
He's number 4.
And stuff. So I was like having difficulty with that one because I wanted to start seeing like plastics.
On the registration.
Yeah, I really be really careful with that one because you got those like it's cool with like other plants and stuff like out in like nature stuff, but like there's like a certain plant, I forgot what it was. I don't know if it's a shrub or a tree that is actually detrimental to the world. So it was like you can't just let that one research and cause a problem. So I was like okay. So I got it. I forgot where I got it off of online, like the exact source, but that's where I got it. It was I got it in a different country. And then finally, yeah, it was just like, I followed a couple of YouTube videos and like, yeah, it's super easy. And I was like, I did something wrong multiple times. So, because it didn't launch, and I was just like, so I've been getting more tracker down. So I'm trying to do the
we're just talking about
so I was like, I'm doing something right. So
the next
I think what I saw. Oh, then I saw people like they were doing it in microcultures. Like so,
Well, I got your
so I
message
guess
of like 2 weeks back, and I decided
that's
to
just
come here
for
to
I don't know if
connect
that's the mycelium
off of
stuff that
the
you would call that. Like
mycology
it's in liquid form
software.
and it starts to grow. This thing you can break it up and shake it up and
And I'm
make the syringe and then
aware that there's 3 more people left that we can play on coming.
is convert that over into like a jar with a bunch of plastic pills and it actually yeah.
Oh, I was thinking after that. Otherwise, I'm going to have to put my finger right
Absolutely fact check me on this one this time. So I think he had it up in like 3 weeks to like a month. So we started seeing signs of it taking off. So that was like
what's it called?
so I don't know if it's
Just
all
open up
plastics,
the science report.
and dormant drugs. So I thought that
Makes sense.
was
Nice to meet you.
Hi.
How are you doing? Yeah, I'm doing okay. I noticed that you have not OBS running for YouTube, Twitch.
Yeah, for YouTube.
Oh, okay.
I imagine ones that are
Oh, it's your first time using it?
Well, we use it every time, and every time it's different.
Right, because every mic or
Just
your
because every
different.
time it's different.
Okay.
That's weird.
We got to see y'all again.
Yeah, it's good to see you again.
I've been meaning for that. I thought it'd be a fun night to get yeah, same with the blue oysters. I was hearing like taking a highway.
Is there anything you missed that you were
Or it was like blue oysters and something else for introducing
strings?
like suck it up on the soil and then just turn it into
All of us paid for it.
Oh, that's nice.
We're getting there.
Did you have to get 1,000?
Did you just say 3 people?
We need about 1,000. And we need about 300.
Oh, 300. I only heard 3.
Like we're getting there. We're almost at 100.
If it's only 3 hours.
Yeah, it's only 3. I already have 4 YouTube accounts.
I don't know if they sign up for actual use cases where
The thing about
substrates that you only pasteurize. So you can just simply boil this. You're using a lot less energy, a lot less technical equipment, and it's a lot safer to boil a substrate rather than have 15 psi vessel. That's essentially a bomb in the sterilization process. So it's a lot easier if you can pasteurize your substrate to produce mushrooms than to go through the sterilization procedure. But once you have your fungus inoculated, you let it grow through the substrate, what's going to occur is that it's going to have essentially starvation at some point. This is one of the cues that causes the switch from the mycelial form to the mushroom producing form. The fungus is running out of nutrients, and if it were to be able to think by anthropomorphize, it needs to produce spores. It needs to produce these environmentally hardy forms because it's detecting that the environment is becoming inhospitable. There's no more nutrients left. Therefore, producing spores that can survive in the environment until nutrients can come back to the environment. So that's only one of the signals. A couple other signals are environmental cues. Like I mentioned, the mycelia is growing within the substrate or underground. Only when it gets to the surface should it be producing a fruiting body, because that's where it can disperse its spores a lot more effectively. So you have underground, you have no light, you have a buildup of carbon dioxide, and so when the mycelia gets to the surface, that buildup of carbon dioxide can disperse. And so a drop in carbon dioxide is another cue. Light for some species can be a cue, and then also a change in temperature, and then a rise in humidity can be cues. So these are the kinds of things we want to replicate inside of the Roman champignon, de Paris, the portobello. These are all Agaricus bisporus. They're all the exact same species. They're just harvested at different times. You just take a young mushroom, you have a button mushroom, you see these on salads. You take a much older mushroom, you have the portobello, you see these on portobello burgers and things like that. All other mushrooms are considered specialty. So lion's mane, chestnut, oyster, shiitake, these are some of the most common ones that we grow. And when you look at how many mushrooms are produced in the US, you see about 2% of specialty mushrooms produced, where the vast majority is this Agaricus bisporus. And they're out of aspects that we focused on when we were trying to develop our first USDA grants at KSU. We applied and were awarded 2 grants over the course of several years to develop effective technologies and also look at using Georgia agricultural waste streams to grow mushrooms. So taking these low to no value agricultural waste materials and growing human food products from those. So these controlled environments started off with this small incubator in our lab, controlling CO2, temperature, and humidity. And with our USDA grant, I was able to take this 20-foot dry shipping container and sort of retrofit it with the software I developed and hardware I was developing to produce an environment to grow mushrooms. And so some of the modifications are using closed-cell spray foam insulation to insulate it, producing an airflow system where we're taking in fresh air, filtering it, and that allows us to exhaust out the CO2-laden air that the mushrooms are producing. A humidification system to keep the humidity high, and then an air conditioning and heating system so we can regulate temperature. And so this one has a PVC frame with a plastic chamber on the interior. This has served us since 2018. We are continually doing work with mushroom culture. These beautiful Georgia cotton balls, a little bit of cotton. And so this is called cotton gin trash, or the push in the industry is to call it a more friendly name like byproduct. But this is sort of what it looks like. It's sort of a mix of a little bit of cotton and a bunch of these other organic materials. And so there's a huge amount of this produced every year in Georgia, so about 200 million pounds. And this has a pretty low to no value to the farmer. It can be used for roughage for cattle, so it can be mixed with other materials and cattle can eat it, but it's also not very nutritious for them. But it's extremely rich in cellulose, and mushrooms love to break down cellulose and feed off of it. And so with experiments where we were looking at different formulations, different agricultural byproducts, we found that sort of a 50-50 ratio of cotton gin byproduct and peanut shells was ideal for growing oyster mushrooms. So we grew a lot of oyster mushrooms on this material. Okay, so from our first USDA grant, we learned a lot of things about this R&D process of building a controlled environment for mushroom growth. We got another USDA grant to build out these and gave them to 3 mushroom farmers in Georgia to trial for about 1 year to 2 years. And so some of the improvements moving from a dry shipping container to a refrigerated shipping container is we don't have to insulate the walls. They're already insulated with about 4 inches of closed cell foam insulation. They have a stainless steel interior, so they're already food grade. It's also about twice the size. If we go back, actually, let me go back, sorry. I know you're trying to get the slides here. In the original version, we had a shaver. So with that closed cell foam insulation that I put in the Gen 1 dry container, there's a lot of crevices, and that's why I put that secondary chamber on the inside. Is because if you applied a high humidity environment to that closed cell foam insulation, you have a lot of different molds growing on it. So this is a very nice food grade, smooth surface that can be cleaned, and so there doesn't need to be this additional chamber, and so you can maximize the grow space of this. And so for a larger environment, I also have a larger ducting system. There's a continuous circulation that filters the air, and then all the air coming in is also filtered. I did move the control panel on the outside just to keep it out of the high humidity, and similar to the first unit, there's a window air conditioning unit. In the picture I showed, I was using a different air conditioning unit, but eventually went to a window AC unit. And so this is just one of our growers, Gowin Valley Farms, that in this photo at least is growing predominantly lion's mane mushrooms, and they have some of the most prolific lion's mane mushrooms I've ever seen. Okay, so let's sort of go over what you need to grow mushrooms. And I sort of mentioned these, but this is sort of the hierarchy of what is most important, at least what I found to be most important. Humidity is going to be the one condition that is either going to make or break a high or low yield. You want somewhere around 85 to 90% humidity, and if you get down to 70, 60, 50, it's going to severely diminish your yields. Next is going to be fresh air. So mushrooms, like us, breathe in oxygen and they respire CO2. And so also like us, if the concentration of CO2 builds up too much, then it starts to become toxic, and you can start to see reduced yields, or you can even kill the mushrooms. And then temperature is also important, but this is going to vary between species. Even different strains of species, you may find warm thriving strains of an oyster in Georgia, whereas up near Maine you may find cold thriving strains. So it's going to be species or strain dependent about what temperature is going to be ideal. But generally lower temperatures are better. Most mushrooms can form at lower temperatures, and they often will grow denser and have like a better texture. But the same is not also true for warmer temperatures. So for instance, a cold thriving mushroom can grow well at cold temperatures but not warm temperatures, but a warm thriving mushroom can grow equally well, if not better, when it's in warm temperatures and cold temperatures. So that's why I sort of say that that's not necessarily the most important condition. Now, if you were to want to set up some sort of manual environment for regulating conditions, this can be done pretty easily. This is a system sold by Northspore. It's called the Boom Room. And it essentially is a small greenhouse that has a humidifier. And I'll show another component they actually include. But if it were to be fully manual, where there's no automation involved, you have a humidifier that you set the output, and you have a fan that you set the fan speed. And this will keep continuous humidity being pumped in to keep the humidity high, and you'll have an exhaust fan that's continually pulling the CO2 out and pulling in fresh air. And so this is pretty effective, but one of the issues you'll have is if you have different capacities for growing, say you put, if you double the number of substrate blocks in your fruiting environment, you're going to double the production of CO2, and you'll have to then increase the fan speed, and then because you're pulling out twice the amount of humid air, you then have to turn up your humidifier. And so depending on how much CO2 is being produced, you have to then constantly adjust your fan speed and your humidity production. Now how we can automate this is we can include a CO2 controller right here and a humidity controller. Now the actual boom room comes with a humidity controller, but not the CO2 controller, because these CO2 sensors are really expensive. You're probably paying the same amount for your CO2 controller as the entire boom room itself. But what you can do is you can add these two controllers that have a humidity sensor and a CO2 sensor that you put into the environment, and it will turn on and off your humidifier and fan to be able to regulate those different conditions. So for instance, with your humidity, you have a certain range that's acceptable, let's say 85 to 90. detects the sensor that the humidity goes below that. It turns on the humidifier, and then when it goes above that 95%, it'll turn off the humidifier, effectively keeping it within that range, completely independent of what the fan is doing or what type of airflow is occurring. So with these two controllers, you can regulate that humidity and CO2 and have a reasonably well-working controlled environment. Now, there are some issues when you get to scaling. So if you wanted to move to a larger environment where you're trying to, say, produce at a commercial scale, this is where, it's sort of what does commercial mean? I sort of see it as whether you're able to support yourself, whether this is a part-time or full-time job or beyond. That's when you're getting into commercial scale. When you're looking at about 20 hours per week or more devoted to mushroom cultivation and sales and everything that goes into getting mushrooms to market and making sales. You're going to have some issues when you start to go through this scaling process. So you have to look at the size of your environment, and this is going to determine how reasonably well you can mix the air or homogenize the air. So you have to then size up your components, your humidifier, your fans. You have to then start to think about dry spots or measuring different points within your environment. It's going to be crucial when you start increasing the size of your environment. Insulation, temperature control, and thermal bridging. So once you start increasing the size, you're going to need to increase the capacity at which you can cool and heat. And then you're also going to have to consider the insulation value of your, and then certain thermal bridging. So if you have a structure that's out, outside, thermal bridging is where you have, say, a stud that is exposed to sub-zero temperatures. It's going to cause that stud to become cool. And if that stud makes contact on the inside wall, you'll have these areas on the interior that you can see getting very cold. And so how this is an issue is that in an environment that is extremely high humidity, cold causes condensation. So anywhere you have this thermal bridging and these cold patches, you're going to have moisture being pulled out of the air, condensing, dripping down, and creating pools of water. So there's a couple issues with that. You have, one, the humidity dropping when you have condensation forming. Two, it's a slip hazard. And I think one of the most important ones is now you have an area where different contaminated microbes can grow. So where you have pools of water, you have bacteria and other fungi starting to grow. And so contamination is a pretty big consideration inside this environment. You're trying to filter the air and do the best you can to keep these contaminated microbes out. Now with these environmental controls, the two that I mentioned, there's a couple different companies that make these very simple controllers. Inkbird is one of them. But with all of these, or most of these, you're going to find they come with one sensor. And the issue with working with one sensor is you don't know when that sensor is starting to become fouled and that measurement starts to drift from what it actually is. When it's new, it's going to be measuring what the true humidity is. But if you have condensation forming on the sensor, or if you have spores landing and fouling the sensor, then it's going to start measuring either higher or lower than it actually is. That's going to mess up its ability to regulate. If you have multiple sensors, it's likely you're going to see fouling occur on one or a few of them, but not all of them. You can start to detect drift between your sensors, and then you can do some testing to find out which sensors are bad and replace those sensors. Which comes to the next point, being able to replace them. These cheaper controllers, you're not able to replace your sensors, so you have to buy an entirely new controller, or you're getting down to having to desolder, take apart the controller, and solder in a new sensor that's the same chip, which is not always in your DIY tool belt. And then you also have multiple controllers that are acting independently. They're not acting in unison, so they're often battling each other. What you usually see is an exhaust cycle will cause a drop in humidity and a rise in temperature. Particularly in the summertime when you're bringing in that warm, cool air, that warm, dry air. And then you have the humidifier and the AC kicking on after the fact. So you already start to see that drop in humidity before that controller can counteract it. Whereas if they work in unison, if you can anticipate an exhaust cycle occurring, you can preemptively turn on the humidifier so you don't see that drop in humidity as strongly. So you can prevent the negative effects of your mushrooms experiencing that drop in humidity. And then for some systems you have this stability or resilience issue. So for some control systems that are running, say, a full operating system, back in the day with other file systems, it was a little more common where if you had a power outage, you tried to boot your computer back up, and it said file not found or operating system not found. The operating system can't boot, then the control system can't boot. So looking at different systems that are more or less resilient, and I'll get into that when I talk about some of my software, is an important aspect as well. And then you have some of these non-environmental regulation considerations, like your sales channels. As you're scaling up and producing more mushrooms, you have to find more businesses or people to buy your mushrooms from you. And then also there's certain limitations where when you're small enough, you don't need certain certifications or licenses. And then once you meet a certain capacity of mushroom production or sales, then you sort of have to show that you have these safe working environments to be able to produce produce, sinks and things like that. So it becomes a little more complex outside of the growing environment as you increase the capacity of growth. And so some of the negative effects you can see from these issues is you can have reduced yields, you can have contamination, you can have disease, and other effects from contaminated microbes growing on or around your mushrooms. And then you can also have these weird phenotypes of growth where it may or may not necessarily affect the mushroom itself, have a taste or a nutritional value, but they look very unsightly and you're not going to have customers wanting to buy them. So these are oysters that are probably a little past their prime, but they're also curling up. Very strange growth. So this is the last part I'm going to get into, and this is sort of the advanced automation control. And I'll talk a little bit about some of the software I developed. There's this software suite called MycoLogic. This is something I've been developing really around since 2009, but I haven't really put a lot of effort into it until starting around 2013. So this is free and open source, meaning you can download it, you can go to that website, you can put it onto this $50 computer, a Raspberry Pi, you can connect sensors and output devices, and you can use it, you can modify the software, you can use it even commercially and make money off it. I don't need any money from it. The GPL license that's released on really only requires mainly that you attribute me as the author. But it essentially takes inputs or sensor measurements and outputs or rather controlling different devices, and then functions that couple those together, and we're called feedback loops. So like I mentioned with that simple controller, that's considered what's called a bang-bang controller, where when it is measuring under or over a certain value, it'll just turn on an output until that measurement either comes below or above another certain value, and then it'll simply turn it off. And that's good enough for most regulation. That's where you really have the power. This makes it a little better than those simple bang-bang controllers like made by Inkbird, because you don't have the power to, if you learn a little bit of coding, or even without that, I have an interface where you can do visual coding with this and have things work without having to learn coding. But this gives you the possibility of creating new functionality that didn't otherwise exist. So I'm just going to go through some of the projects that people have used this for. I think there's around 1,000 active systems around the world. People are using this to grow different types of food or age tobacco, cheeses, smoked meats, produce smokers, of course grow plants, grow mushrooms. There's a lot of different uses. These are just a couple projects that I highlighted here. This is one of the very first people that I worked with to help refine the software. He was starting a sort of a business with his kids to show them how to learn hardware, software design, how to manage money. And they were breeding these rare phenotypes of corn snakes and selling them to pet stores that they can resell to different people. So he was using this in a simple aspect of regulating temperature. So he was incubating the mice he was feeding to the corn snakes, the snakes themselves, and then the young, so that they could hatch and have the highest likelihood of increase the number of eggs hatching. There's a lot of people that grew mushrooms. This is just one. This is Jocko from Russia. He was growing lung oyster. I'm not too familiar with the research that was going on here, but Victor was measuring a whole bunch of different metrics, scale, aquaponics, the effects of different treatments against white nose syndrome, the fungal disease of bats. And in order to replicate a more typical roost environment for bats, I was regulating humidity. So I was measuring humidity, and I was using these ultrasonic misters to be able to raise humidity to that level. This is an artificial apiary where we were think we had about 30 to 50 honeybees that we were testing whether we can keep them alive for a long period. I believe this experiment went for about 25 to 30 days before the bees died. And this was an initial study to be able to then do experiments with the fungal pathogen Ascosphere apis. It's a pathogen of honeybees. The western honeybee, Apis mellifera. And so we have honeycomb here, we have bee pollen, and we have two sugar water sources for them to be able to feed on. What's interesting is you can see they sort of go into dormancy at night, and then right around morning into midday, you can see their activity, sort of increase to maximum. What's interesting is that this is in complete darkness. I actually have infrared light to be able to have this camera view, but this is their natural circadian rhythm. Even without seeing light cues, they stay on for every single day for this entire 30 days, this cycle of going dormant at night, 4 a.m., and right around 9 a.m., they get really active, and around noon, you can see there's 9 a.m., they start becoming extremely active again. So that's also pretty interesting. So this was inside a lab that had constant fluorescent lights on. It was also mostly light tight, so there was no way any sunlight was actually reaching us when to have that as a key. Implications with disease management, and so this is a method where you can change the temperature over time. So we have a temperature sensor in the water bath, a heat plate on the bottom, and then the heat plate would energize and de-energize, and we would measure the temperature of the water. And over time, we would either raise the temperature midday to simulate what would occur naturally, where the sun would be high noon, creating the hottest part of the day. Here it is, just one test around about 45 degrees, and then the lowest part of the day about 25 degrees. We're testing what would be simulated in Africa. And then this is sous vide, which is French for under vacuum. So this is a way of cooking using temperature-regulated water baths. So you can reach the perfect internal temperature for your food, be it a meat or a vegetable, without actually going over temperature and overcooking it. So you can have a perfectly rare meat, and then you sear it on a scorching hot cast iron pan, so you get that perfect caramelization, but perfectly rare, safe to eat inside. And then if you go to my YouTube, you search my name, or you go to my website, you can see some of my latest projects related to automating hydroponic systems and automating mushroom cultivation. So you can go through the video, it's a little more entertaining, and accompanying that on my website is a build article, 20, 30 pages long, going into detail on how to replicate an automated, fully automated hydroponic or mushroom cultivation system if you wanted to go into detail with these integrated control systems with my open source software and very cheap, inexpensive controls hardware. So that sort of like brings us to the end, where most recently I formed a company called MycoLogic. We were established with my co-founder, Chris Cornelison, who's also my colleague in the laboratory at KSU, in late 2021. This is when we started really developing the third generation of our fruiting module. And so we stayed with the 40-foot shipping container shell, but we had a lot of improvements to that, where we're now using actuated dampers that we have complete control from 0 to 100% opening. We're still using speed control fans, but what this allows is to have very precise control of air coming in and coming out. And this is most important because at these larger scales, any saving of waste is important. So being able to only remove the amount of CO2 that's being generated by the mushrooms allows you to use less energy in cooling and humidifying, and so you can maintain those environmental conditions much closer to what you want them to be, and you also reduce the cost in energy expenditure because you're not heating or cooling or humidifying any more than you absolutely have to. If you look at some of the photos, you can see now we have a solid floor, whereas that reefer container had a slotted floor that was incredibly difficult to clean. We have a lot more lighting. We have about twice the amount of lighting, so it's incredibly easy to see what you're growing. The walls are bright white, so you can easily clean them. Instead of that 18,000 BTU window AC unit, we now have a 36,000 BTU heat pump that can both heat and cool. And we do have a demo farm in Rochester, New York, where you can see here we are almost at max capacity. We've had it in operation since March, and you can see some oyster mushrooms on the left growing and lion's mane on the right. And just to show sort of the evolution of the control panel, you can sort of see this is one of the first control panels that I built to this scale, and you can see it's sort of a rat's nest of wires. There's no labeling, and that's largely because it was an R&D control panel where there's a lot of changes going on. And as the years goes on, I've learned a lot. I've almost become an expert at building control panels. You can see there's now a lot more organization. There's wire ducting, there's wire labeling. We have complete wiring schematics, so you can hand it to an electrician and they can reasonably understand and replace any component. So this can be done, without having myself have to do a house call or fly across the U.S. to be able to fix something simple. And then there's sort of like, you can see the evolution of features that have been added. One of the most important facets is that these were using just Raspberry Pis, which is that full operating system. Now, if you have a power outage and it has a file corrupt, you may see that your operating system doesn't boot when power is restored. And if your operating system can't boot, then your regulation system stops working. Now, I've started using PLCs, or precision logic controllers. These use microcontrollers, which you find in all sorts of household electronic devices, such as microwaves. We know that if the power fails on a microwave, it comes back on, it just loses the time. It doesn't prevent the operating system from running and you able to use it. So the core functionality of all the regulations is controlled by the PLCs, but there still is a Raspberry Pi that has the web interface for you to view data, remote monitoring, reconfigure remotely, and the database to store historical data. But we now have a separation of the functionality where all key regulation aspect is on the PLCs. So it severely increases the reliability. So even though you may lose the Raspberry Pi if it were to have the inability to boot, you wouldn't lose the ability of your growing environment to maintain its conditions. So the most important aspect is keeping your mushrooms growing, and it's able to do that even in these situations where you might have power failures caused data corruption. And then the evolution of the shells. So starting with these smaller 20-foot shipping containers, these dry containers that needed to be insulated, moving to reefer containers that had the unideal floor, to these purpose-built refrigerated containers that we've now brought back the vestibule. You can see there's maybe like a 3-foot vestibule here, but now we have an 8-foot staging area where you can have, you have your hand sink if it requires that for certifications, or just general washing hands. You can have areas where you put lab coats or shoe covers or gloves. And so it's a general staging area. So you can come into this area and just make sure you don't bring in any insects. You can put on all of your PPE before going into the growing area. And we also offer a version without the vestibule. So there's two different areas of thought, whereas if you just have 1 or 2 units, you may want this vestibule because if you don't want to be standing in the rain bringing mushrooms out, you want some sort of staging area. But if you want to build a very large, and you have 4 rooms, where you have a door in the front and the back. You have a substrate mixing area with a ribbon mixer where you can mix, hydrate, and bag your substrate. The next room is where you can pasteurize or sterilize. And then the next room is a laboratory where you can work with your cultures in a sterile environment, and then use a laminar flow hood where you have sterile workspace to be able to take your sterile substrate that you just, or more people working to produce at that type of scale. And this doesn't really cater to somebody who has only a little bit of experience growing mushrooms or no experience. That's a big leap, and it's also a big capital investment. This is sort of to reach the customer demographic that wants to sort of ease into it. Lower investment, so lower time investment. And this, with our first product, we're sort of targeting an 8 by 8 foot room, but using these insulated panels, they're modular, so we can pretty much build any dimension that you want for a grow room. And this we're sort of looking at releasing for about 15 to 20,000, where those other products are anywhere from 130 to 160, 70,000. So this is sort of allowing you to sort of ease into mushroom production at what would really be considered the smallest commercial scale production. If you were to maximize the production in this 8 by 8 grow room, you'd be looking at a couple $100 per month. So this could be up to about part-time 20 hours per week of producing, harvesting, and going out and selling those mushrooms. I just want to show some of the people and organizations that helped us, do all these things. There's a lot more, but these are the main ones: Georgia Research Alliance, Georgia Department of Agriculture, Kennesaw State, and their incubator Hatchbridge. And then we also won a pitch competition with Grow New York that gave us $250,000 to sort of propel this into where it is today. And then also you can find my company at MycoLogic.solutions, and you can email us if you want any other information about any of our product offerings. But this is really the end of my talk, and if you have any questions, I'm happy to answer or discuss really anything related to mushrooms. It doesn't have to be related to the company at all. So, thank you.
All right, thank you. Does anyone have any questions?
I'm gonna frame it as a question, but it's more of a comment. Do you feel like how sensitive mushrooms are to the conditions that like the Myco software you built are acted as a pressure for your software being so good for other applications?
I don't know if I'm understanding the question.
I understood that Mycoto got used for like other growing other things, and do you think that like how sensitive mushrooms are made it so that we needed to fine tune that software?
Yeah, so different mushrooms have different varying sensitivities to environmental conditions. Some can grow, if you throw a bag in the back of your closet, they grow just fine. Others, you need to have very tight control of your conditions. I think what really made Mycoto popular is that it's sort of like a Swiss Army knife. There's a huge number of sensors, more than just environmental conditions. There's weight and angle and acceleration. So it has a lot of applications in physics, and there's outside of even growing things that people use it for. And just how easy it is to start using it. You can buy a $50, or Raspberry Pis you can get down to like $10, $15. This software will run on. You can buy a $5 sensor and a $5 relay, and now you can create your own feedback loop controlling devices with less than a $50 investment and just a few hours of your time. And so it really started with the hobbyist, and then once it started appearing in say Google searches for people mentioning it, it was able to be found by a lot broader audience, and people in universities started taking on, because if you're a grad student working on a project where you have a limited budget, you're trying to maximize what you can do with the smallest amount of money. So if you can say, I can make this environment that can grow plants for whatever for $50, that's extremely valuable. Yes.
So this might be more of a difficult replacement cycle. If you're using them for like a small scale, like you were showing with the little rotating and stuff for that environment. What do you find generally is like the replacement time for those sensors before you start getting, I forgot the exact term, but we're starting to stray from the measurements?
Yeah, so for humidity, just for an easy example, it really depends on how you're taking care of your sensor. So if you're putting it into a housing that doesn't get a lot of airflow, it's going to have less exposure to spores in the air. If you're constantly filtering out the spores, it's going to have less exposure to those. If you have really tight control of humidity and you don't allow it to get to 100% humidity, where when you have 100% humidity get reached, even by accident, condensation forms everywhere on all the surfaces, even your sensor surface. So if you don't even allow it to get to that high humidity, you don't allow that condensation or moisture to form on your sensor surface. So the more you can do to prevent the sensor from fouling, the longer it'll last. I've had $10 sensors last over 2 years and be almost as accurate as they were when you first put them in. I've had other sensors that you put in and a few months later you have to replace them because you did something dumb and you sprayed it with a hose directly and got water on it. Because once moisture gets on that very sensitive sensor surface and dries, it's leaving some sort of film, and it's not going to be nearly as sensitive as it was prior to that, or as accurate.
With like sensor cleaning, can that be like salt with using like alcohol or anything like that in spreading it? Or?
So for like humidity sensors, you're talking the sensor surface has to be seen with a microscope to actually see how small it is. And there's a whole bunch of different sensors, but if we're just talking an example with this humidity sensor, you just have to replace it. There might be a way of stripping down to the bare sensor and maybe doing a solvent bath, but I At that point, you really need to be talking directly with the engineers that made the sensor to know what's safe and what to do. But at that point, they're just going to tell you to buy a new sensor instead of investing the time and telling you to clean, the sensor because they want to sell more sensors. But there's probably the ability to do that. But a lot of these sensors also have their own filter membrane, so you would have to get in with a tweezer, peel that membrane, get to the sensor surface, figure out a way to dip it into a solvent bath. So it's not easy, especially when you get into, say, CO2 sensors that use infrared that have a sealed chamber. If you were to rupture that chamber, it's under a vacuum with an inert gas inside of it, and then a calibration gas, because it has two IR beams, one with a calibration gas, so it knows what the signal with the IR sensor should be for a particular concentration of CO2 or whatever gas. And then it has the sensing chamber with another IR beam that is actually taking a measurement. It's comparing to the calibrated chamber. So there's a lot of iffiness of taking apart sensors, and when you put it back together, is it able to be calibrated properly? But a lot of times with these more expensive sensors, you can go through a calibration routine. So even if it starts to drift, you can create an offset. So let's say it's measuring 200 ppm CO2 greater than it actually is. You can just tell it offset at 200, and now it brings it back down from 700 to 500, and it's still relatively accurate now, even though it may have drifted over the course of 2 years. And that's often what the calibration does, is that you tell it this is what the CO2 is right now, and it'll calibrate to returning a measurement of what you tell it is. So there's going to be a point where it drifts so much that it becomes wildly inaccurate, but at that point you should just be replacing the sensor. Unless you really know what you're doing, you can maybe try to clean it.
So let me ask one more question. I'm not trying to impede on anybody else. Sorry. With the software, is it multi-platform? Can you use it like Linux, Windows, and
it's only Linux, so you can run Docker in Windows, Mac, Linux, and that creates a Linux environment within whatever operating system you're running. So Docker runs across every operating system, but it does require a Debian-based Linux operating system to run. But it's really just one command. Once you install Linux, you give one command, and it will install MyCoLogic, configure everything, install the database, and then they'll just give you an IP address to connect in your web browser, and then you'll have a graphical interface to configure it.
Awesome. Thank you.
But really the benefit of using, say, a Raspberry Pi is you have what are called those general purpose input-output pins, and that's where you can connect your sensors and output devices directly to those pins. Whereas, say, on a Mac or Windows PC, you don't have those pins broken out. Like even if you have access to your motherboard, you usually don't have those GPIO pins to be able to connect sensors or connect output devices. So it's good to experiment on a Raspberry Pi, but eventually you're going to be moving to maybe using something a little more purpose-built. There are hardware platforms that are a little more robust that aren't running full Linux. But at that point, you are probably deep into automation at that point. But before then, running it on a full Linux system is, especially for home use and hobbyists, it's perfectly fine. Because if you have, say, a power outage that causes data corruption, you can just pull the SD card and put a new one in, and that's your whole operating system. So it's good to have a complete backup of the SD card that runs it. It's where if that happens to occur from a power outage, you can just pull the SD card, put a brand new one in that has a complete copy of what was there before, and you're back up and running.
Cool. Thank you.
Yes?
Like for the substrate, what kind of
Pre-colonized, and you're getting the blocks ready to fruit. So you really only have to worry about producing the fruiting environment. You get the blocks, you cut a slit in the plastic, and then put them into your fruiting environment. And then because now the mycelia is in an ideal environment to fruit, you'll have mushrooms start to grow out. So you're really only having to worry about that fruiting stage. And then once you get familiar with that and you want to maybe increase your margins, do we see farmers going into actually producing a lab or a lab-like environment and sort of returning those margins by producing their own substrate, or maybe buying pre-sterilized substrate but then doing the inoculation and then getting familiar with that part of it before easing into mixing and hydrating and sterilizing their own substrate and then doing the inoculation. So there's a way you can sort of ease into the full process to sort of maximize your profits and increase your margins, but sort of recommend that don't bite off more than you can chew and just start with one part at a time and then slowly learn and go through the trial and error of figuring out what works works and what doesn't, instead of just saying, I want to go, completely from spore to mushroom, the entire process, without having actually ever done that before. Yes?
At the grocery store, you have organic mushrooms and inorganic mushrooms. And so if I understand the cultivation process right, it seems like cultivating is mostly organic, right? Or what's
Yeah,
the
so
difference?
I have a lot of opinions about organic mushrooms, and so the question was the difference you see in the grocery stores with organic versus non-organic, and how you would essentially produce an organic, or do you even need to produce an organic mushroom? So to really understand this, I sort of have to talk about plant physiology. And so plants have a vascular system where if you put a nutrient in the soil, that chemical, that nutrient, will physically be taken into the roots and pulled up through the vasculature system, and then put into a leaf or a fruit of the plant. And it's vastly different from how mushrooms are produced. The mycelial network of fungi produce exoenzymes. The mycelia secrete enzymes that will break down complex materials in the environment, and that's actually how plants get their nutrients. Plants rely on fungi to be able to even survive. So the fungi produces exoenzymes, breaks down the organic matter in the environment to essentially smaller building blocks or chemical components that the plants pull in through the roots up into through the vascular system. Mushrooms, on the other hand, will absorb those basic chemicals into their cells, and then they will grow new cells. So the mushroom that's formed is taking bare chemicals, like the building blocks of chemicals, and then building more complex carbon sources or sugars that make up the mushroom. Whereas plants can take in a more complex molecule like a pesticide. And so that's the issue, or the concern with plants, is that if you spray a pesticide on plants, even if you try to avoid hitting the root system, when it rains or when water hits the plants, it drips off the leaves, goes into the soil, and the actual very complex molecule that is the pesticide can be brought into the plant and deposited into the fruit that you then eat. Mushrooms will break down those pesticides into small 3-4 carbon compound chemicals, which will then be taken into the cell, and then those will be building blocks to create more complex carbohydrates that make the mushroom. So we've done studies at KSU that take substrate that's heavily contaminated with pesticides. We then grow mushrooms on them, and then we take that heavily contaminated substrate, we take the post-colonized substrate, and we take the fruiting body itself, and we sent those over to a lab to analyze for pesticide residues. And we found that, of course, the substrate is heavily contaminated with pesticides. The spent substrate that mushrooms have been fruited from have pesticides detected at a much smaller level, and this is what we would expect because the exoenzymes are breaking down the pesticides. And the fruiting bodies themselves, of the ones we've tested, there was no detectable pesticides in them at all. So even though your substrate may be contaminated and not considered organic, there are a lot of people that would certify your farm for organic, that would understand this process, and they would be able to label your mushrooms as organic, even though the sources that grew the mushrooms are not organic. And so this is a sort of contested topic about between the certifiers of organic, is that some will say you need organic substrate to produce organic mushrooms, and obviously that is not necessarily true in all cases, and it's also much more expensive to source organic materials than non-organic. And comparing that to the people that would certify organic that say you don't need to have organic substrates, that or mushrooms are naturally organic regardless of what substrate you grow on. So that's my take on it. I mean, you can come to whatever conclusion you want, but really depends on
I
the.
struggle with this.
Yeah.
Like the grocery store, I'm like, what am I paying for?
Yeah, with plants, it's a reasonable concern. With mushrooms, you have less of a concern, I would say. There's still the ability to cross-contaminate, whereas if you touch the substrate, you may inadvertently move chemicals from that by touching your fruiting body afterwards. But the transfer naturally from the substrate to the fruiting body is probably very unlikely because of how the physiology is different between plants and mushrooms. Yes.
I was just curious, when you guys did that study, what species of mushroom did you use?
We're using oyster mushrooms.
Oyster mushrooms. Okay, cool. Awesome.
Yeah, oyster is a very fast-growing mushroom. So So, and it's also very hardy, so you can get away with just pasteurizing or killing a large amount, but not all the microbes in the substrate, and it just grows so fast. That's one of the reasons why you don't have to kill everything, is that when you inoculate an oyster mushroom, it just outcompetes everything. So it's really easy to work with. It grows fast and very large fruiting bodies. But it's also known, we were talking about this earlier, its ability to break down really complex hydrocarbons is used in bioremediation of oil spills and things like that. So we saw it as a natural candidate to test its ability to break down different pesticides.
Cool.
Yeah.
As far as like the use cases that you guys are usually getting after, is it usually like for cooking or for other commercial uses?
For the mushrooms that are grown,
Yeah.
it's usually culinary mushrooms. This also does overlap with medicinal mushrooms, because some culinary are medicinal, like lion's mane, you can cook with it, but it has medicinal properties. And then there are some that are just purely medicinal, like reishi. It's a very hard, woody mushroom that you really only would make tinctures or grind the powder and use it as a supplement. But it usually falls in the realm of culinary and medicinal mushrooms. That there is another area of growing where there's different compounds like psilocin and psilocybin that are in sort of research phases. And so those are a little less common. You get a DEA license in the US to be able to research those, but there are groups that are trying to grow mushrooms, in fact, like psilocybin producing mushrooms to be able to do the extraction, to be able to even do those types of studies with like PTSD and other effects with neuro effects. But yeah, it generally falls with culinary is usually the most types of mushrooms for growing. Then you have medicinal, and then you have psychedelic mushrooms. Yes.
Just to double back on the study that you were talking about with the pesticides and growing the soil and stuff, is that something that's going to be used for like agricultural farming and like soil remediation?
I'm not too familiar with remediation practices using mushrooms. I know that people have done it on small scales, like they've experimented with it in farms, but I don't know of any widely used commercial products. There just really isn't a lot of money in bioremediation, so you don't see a lot of research going into doing that. But there are a lot of people that are doing their own experiments on their property. If you're familiar with Paul Stamets, he's pretty big in the mushroom world. He's done a lot of experiments. He's not formally trained in mycology, but he has a huge publication record with remediation. Particularly with fecal coliform concentration in this body of water. And so he's done experiments where he's injected oyster mushrooms into substrate beds that he's dug. It became a natural filter for the runoff from the fecal coliform that are coming through. And it can be sort of a trap for the organisms, but largely it is the mycelia of the mushroom breaking down those compounds as it goes through these beds before it goes to the water. By the time that chemical gets to the water, it's already these much smaller chemicals that are no longer a concern.
Thank you.
Great. One more question?
Okay, I'm the last
You're the last question.
Okay, okay, I gotta make it good then. Would an increase in nitrates cause the mushrooms to grow in population or make it worse?
It really depends on the mushroom and what your starting substrate is. So when we look at different agricultural waste materials, they have different carbon, nitrogen, phosphate ratios. And so that's why we need to create different formulations, so that we're trying to find the ideal CNP ratio for the particular mushroom strain. So if we were to just use 100% of a particular substrate, we may get X amount of mushrooms that produce from that. But if we were to supplement, say, 50% peanut hulls into that, we may get X times 2 or twice the yield of mushrooms, simply because we're providing a better nutritional source for the mushroom. So that's the whole idea between creating formulations, is finding what you can add to your substrate to be able to increase that yield. And so there's a lot of different adjuncts you can add. There's lime, there's different nitrogen sources. If you're using things like compost, if you say have a compost produced from leaves, you may add spent coffee grounds to be able to add a nutrient that's lacking from that high leaf content compost soil. So there's no real answer like, do you need just nitrogen? But it could be that you have too much nitrogen and you need to add more carbon and phosphorus to be able to make a perfect balance for that particular strain you're growing. So some are more sensitive than others, and one formulation of ag waste may be ideal for one, but completely terrible for another. You may have to reformulate it to be able to find that perfect balance of nutrients for that other strain.
Thank you very much.
Thank you.
Awesome. All right, let's give Kyle a huge round of applause. All right, we're Science for Georgia. Thank you for being here tonight. Thank you to Mutation Brewing for this lovely space, and hope to see you at an event anytime soon. Thanks, guys. Have a good night.
And if you're interested in any merch, we have some t-shirts and other things.
All right. Great.
How's that?
It's awesome. That was so much fun. I really enjoyed it. That was really cool.
Do you know how long it went? I'm just curious about the time. I see a clock with the things here. Is it about 40 minutes? 15 minutes of questions? Hello. Any of our stuff?
Is there like a
So yeah, the funny t-shirts with wrap. We have some of our science comedy shirts. We have our science crawl, but only this one. They're almost out of this one. We have stickers and buttons and
when you start
bags.
how did you put up the control?
Yeah, there was kind of
Or you had to find
That's fine. You can get a postcard, stickers, or buttons. They're $2 a piece. You get 3 of any of them for $5.
So I didn't really get into the controls. It was like more
Do you have any?
I got to play with it.
You can scan our stuff, or you can go to our website. So it's scienceforgeorgia.org.
Exactly that. But it's for
You can
high pressure, high temperature
take a picture of that.
Okay, I found it.
Oh, really?
So the website at the bottom,
You guys
that
don't
can go to we got huge ones on the website. You can find our merch store
Dude,
there.
that's sweet.
But we have a lot more stuff.
Yeah,
The reason why I asked is
I'm not trying to plug anything, but I'm just kind of like, okay, that's awesome. So,
Yeah, sure. Yours are from like
he has a affordable goal
we use
to
Sarah's
get
brings it
maximum
to
funding.
120. No, that's great. We just did we worked with
okay,
50
that's
Line,
awesome.
Indivisible.
So, have you guys
We
tried to do
did
like
a
versions
press
of that? I know that can give risk
last week.
So